Well, while he's doing that, I'll introduce myself. I'm Ben. I've been with FAR for about 3.5 years now, mostly working with herbicides and herbicides, and also branching off into aphid control and aphid monitoring. So oilseed rape is not something I've had a lot to do with, so I'll be learning a bit today, probably more than you guys, but I do know about what I'm talking about today. So one of the things that I've been doing a lot with, let's see if this is going to do anything. Yeah, no, it's not. Oh, I see. Oh, we've got animations on this one, have we? Did you put those in? No. It might have done it automatically. So we're going to talk about herbicide resistance and grassweed control. So those are the things that I've been looking at quite a lot in my time at FAR. Still only sometimes clicking, which maybe I just need to turn around. Yeah, I do. And then we're going to be talking about viruses and also some other diseases, fungal diseases mainly as well. So yeah, if I sort of stand here, I think it should work. All right, so the first thing I just want to talk about, and this is more broadly not just about oilseed rape, is herbicide-resistant weeds. So many of you will have known that we've been doing this work since about 2018. FAR's been involved in quite a large group which is involved with the CRIs as well, looking at herbicide resistance, and part of that was herbicide-resistant weed surveys. Starting in 2019 and going for 5 consecutive summers until 2023. South Canterbury, where we are today, was in 2020. Was anyone in this room involved in those? Do you know if anyone remember people coming to their properties? And I'd be surprised if there wasn't someone here, but yeah, we went to quite a few places. Of course, it was during all the COVID nonsense, so it was quite an unusual time. But so we've finished the 5 years of surveys now, and we've got all the results. And what we've found is that Group 1 and 2 chemistry, which is Group 1 is the FOPs, DIMs and DEMs, and Group 2 is the sulfonyl ureas, and that's things like iodosulfuron and chlorsulfuron, so grassweed control products. And what have we got next? Yeah, so South Canterbury in particular was quite a hot spot. I'll show you a map of that in a moment, but in particular, grassweeds were the thing that just kept on coming again and again. It was something that had been identified as a risk. We knew that grassweeds were potentially the biggest problem just from some data analysis and looking at overseas, and sure enough it came true when we looked at the results from the surveys. And South Canterbury was particularly a hot spot for herbicide resistance. So this shows a map of all the places that got surveyed. The ones in brackets were just from maize, which was a separate survey which AgResearch carried out, but I threw them in there just to complete the picture. Colour-coded, green wasn't so bad, red is the worst, and you can see right here where we're standing and sitting is the place where we had the most herbicide resistance. The number there, by the way, 71%. 71% was the number of farms that we visited, which was random. The number of farms that we visited that had at least one herbicide-resistant weed. And most of those weeds were ryegrass, particularly in South Canterbury. It was different in Southland. Southland was certainly some ryegrass. Resistant ryegrass was everywhere we looked. We found at least a little bit. But we found quite a bit of resistant poa in Southland as well as some broadleaf weeds. So what's this got to do with oilseed rape? Well, particularly here in South Canterbury, where the rotation options are a little bit more limited, oilseed rape is really important because it's the only broadleaf crop for some people's rotations. I was talking to someone the other day who said his rotation was, and in fact he said it in this room, wheat, grass seed, wheat, and oilseed rape. So a 6-year rotation, oilseed rape for one of those 6. So it's probably not surprising that he has a herbicide resistance problem where he's only 1 out of 6 years he's able to use a different type of herbicide to the ones that are giving him the problem. Oilseed rape's also, as you'll know, a pretty competitive crop, so it can outcompete the weeds quite a lot of the time to some extent. But the main thing is that, as I've already alluded to, because it is a broadleaf crop in in a rotation that might contain mostly monocots, then you're able to use different herbicides to the ones that you'll use the rest of the rotation. So if you've got those grass weeds which are hard to control, which may be resistant to those group 2 herbicides, then you can use something else. So what herbicides am I talking about? Pre-emerge. So I've got 2 here. I'm not sure. But these are the ones that Keith talks about using. We've got a few growers in the room who would use trifluoril and treflan as a pre-emerge. So that is one that people use. Okay, because we were discussing that. Yeah. Because we were discussing this in the office, and we weren't sure. Yeah, here's Andrew. There's a chair there. Yeah, and then there's the other one, ombre. I did write down what. So ombre is used as a pre-emerge. The clomazone by itself doesn't have a particularly good activity on grass weeds, but adding in the allichlor gives it the activity against poa and ryegrass in particular, which are, I think, on the label. One thing I got from Keith's Timely Advice publication is that ombre can be hard on crops if it's used in wet conditions. Any comments on pre-emerges at this point? I don't want it to be completely one-sided.
There's a few using Aphids.
Yep.
I'm just thinking out loud.
Yeah.
Aphids better than allichlor?
So Avidex has got a pretty narrow, well, at least the weed spectrum on the label is pretty narrow. But what we find is that when it's in a mix with other pre-emerges, it can just increase the efficacy overall. It sort of seems to, particularly in dry seasons, it sometimes just gives you that little window before the rains come that gives you the extra control so that when the rains do come, the weeds are controlled. Whether or not, that's our experience in cereals, but I suspect it's pretty similar in oilseed rape for control of grass weeds. Then your post-emerge ones. Clethodim is group 1, and I have said that there's resistance to group 1 chemistry present in South Canterbury and in New Zealand. Clethodim is a bit different. It's still a group 1, but what we tend to find is that when other group 1s have stopped working, quite often Clethodim will still work, will still kill the weeds. However, experience from Australia says that you've probably got about 5 years of use before those weeds also become resistant to Clethodim, because once in that group, once you get resistance to one, you often get resistance to the rest. That's usually the case. But the one that we probably wanted to talk about most today is curb, propyzamide, and that's group 3, so it's a different chemistry. It will kill anything that's resistant to group 1 or 2 chemistry, or both. But it does have a few limitations. So I'm sure I'm not telling you anything new here though, but propyzamide is something that you're supposed to use in the winter. It does require cool temperatures. I think below 13 degrees or 13 degrees and below is what it says on the label. And it's taken up by the roots of the germinating and growing weeds, which means it needs to get washed into the surface. So this is a picture from a trial that we did with propyzamide and cocksfoot. Richard, do you want to say something about this?
Yes, this is 3 years ago now. Cocksfoot near Methven. The main weed in here was Poa trivialis. The main thing I wanted to point out here is we all think about it as root uptake, but actually this trial never got any rainfall after it was applied. I think it was about 3 weeks before we got rainfall. So actually, when it's held up in the leaf area, it does do a reasonable amount of damage itself. Sublethal in this scenario, and all of these cocksfoot plots ended up with the same seed yield at the end of the season. But there is quite a bit of damage that can occur above ground, and we still actually got reasonable control of the POA. It was only at the 1 litre rate that we ended up with a little bit of POA coming through.
Might be too small for you to read there, but that says 1 litre. Yep. This says nil, and it's 1.4, 1.3, 1.45.
And we've done 3 seasons of that now with quite contrasting weather results or weather conditions. So last season, the year before, so 2 years ago, so the year after this, I think we had 130 millimetres within a week of application. And in that trial, the main target was actually Yorkshire fog, so a different grass. And it actually, like, we were worried about runoff and we were worried about leaching, but actually we got quite good control at that point, and absolutely no damage to the cocksfoot. So this appears to be the exception, but I just wanted to put that there to show you that, like, it can do some quite bad or reasonable phyto in those scenarios. So if you're targeting cocksfoot, don't use curb. Basically, we won't necessarily get control. But I'm imagining that you guys are targeting poas and vulpias and ryegrass predominantly.
All right, so we already talked about the first time. So, yeah, so 15 to 30 millimetres of rainfall. In fact, the label, I think, says apply in the rain if you can, which you will no doubt know. And, good thing to point out there that you need to have the soil moisture needs to be below field capacity so that when you do get rain, it doesn't just wash the herbicide off, it's actually washing it into the soil profile.
Yeah, so like that's actually the runoff is the biggest concern in those scenarios, and we do see damage where you get runoff in gullies where it's been applied to the likes of white clover. So 2 years ago in that 130 odd millimetre event, there was paddock of clover that we were doing some trial work in where the next door barley actually died through the runoff of sediment in the gully area. So there are some risks associated with that. That's why we like to see some ability for the soil to hold the rainfall that's expected after application.
Right, which is what you said there. Yeah. All right.
So I managed to find some information or some data that we'd produced quite a number of years ago around curb, and it surprised me actually. Chuck us the pointer, Ben. Yes. So this one, I can vaguely remember it. I don't know if you guys can, but there was 3 years of trials and four different individual trials. 2013, there was two trials in that season. Generally, we think about it as a cool weather product. So this is May applications, and this is July. And in our cocksfoot, this is when we've been ending up the last 2 years putting it on because we haven't had rainfall and it hasn't been overly cold. But regardless of which rate you look at, the means of May versus July, July means across the different years, May means, really does look like May is actually advantageous for putting the product on. So I guess if you took those results, 3 different years, there's different rainfall patterns through those different years, but actually earlier is probably better than later. And when I look through the photos of these trials, I actually think the Oilseed rape plants are doing enough shading of the Vulpia plants in July that we're not actually getting even application. So I think it's probably hitting the leaves of the Oilseed rape, and actually that's an umbrella. And so there was lots of photos where there was pockets of Vulpia just scattered around underneath the canopy, whereas in the early timing it was just pretty clean, obviously, with 100% control.
Do you think that could also be related to the relevant root depth?
Sorry?
Do you think it could be related to the relative root depth?
Of the weed? Yeah, there are possibilities that the weed will have been bigger by this point as well, but we see pretty big, well, I suppose in white clover when we're targeting things at that time, they're pretty large as well, but you're getting really even application because the clover's so small.
This was a trial with David Scott's,
There was one, it might have even been 2 there, a couple at Hursts, and I can't remember whether, might have been 2 at each, I'm not sure. Yeah, it was definitely, Dave's name was on one of the sheets, if not 2. Yeah. Yeah, I found that relative to our other things quite surprising, and yeah, I don't know how many people are planning on curb and have already applied curb.
Too dry, isn't it?
Yeah, that's what I thought, but this season here was actually quite dry. I think it was that one, or it could have, yeah. I have to have a look back through. Sorry?
It'd be dry now, the soil profile from those years, wouldn't it?
There was,
Do you think
I think this one was dry. And I think these ones were quite wet. Yeah, we had a number there that were quite wet in a row, wet winters.
Do you think also good cultivation? If you scratch the top, your grass weeds tend to stay in that top 5 centimetres, whereas if you deep cultivate, grass weeds tend to go around nothing as they get up to.
So, question whether cultivation at planting is, yeah.
Encouraging more deeper roots, grasses as well as
I guess they could have deeper roots, couldn't they, if you've cultivated? I don't know whether these were cultivated or not. That's outside of the information that I found. But anything that's going to be bigger is going to be harder to kill, one would suspect. However, yeah, like usually for hair grass, like this is usually a pretty good timing. It might take a very long time for curb to work, but it usually does get there. But in the photos, it's really bare patch, full of it, bare patch, full of it.
So what's the downside of going early? What's the risk? What are we risking?
Of going early? That's the question, I suppose. I don't know whether Nicole's got any comments there, but like once you hit soil temperature of 13 degrees and have had enough moisture to wash it off the plant, like I say, I'm quite surprised by that data.
Yeah, I would say it's not, don't pin it on a month. Like most months are on the label because typically those are the months where we get those conditions required for application. It really comes down to the conditions. So whether that's May or July for you, that's, you know, when the conditions are right, that's when you should be on it to get the best, I guess, longevity and efficacy out of the product.
I don't know, like you look at the crops where we normally use it and they don't have big canopies, and that was the thing that stood out to me looking at the photos, like clover, lucerne, they're all pretty small at that point of time. And I just wondered if that was actually what was going on there. I got no proof, but we should at least think about it. What was next? Oh. So Keith's got the monitor farms, and
I'll jump in here and talk with Richard here too. So we've obviously got our monitor farm set up, and the curbs, obviously talking from what Ben's showing us, the South Canterbury is quite a hotbed for, you know, resistance to Group 1s, Group 2 sort of thing. So it was interesting, caught up with Nicole the other day, and she's saying the curb's actually quite an old product, so it's not the original research, and that's what, 30 years old. Older than that, so it's yeah. So there's no data showing there's any resistance to curb, but it just, if you try and find any data, it's very old sort of thing. So it's good to have that, and in the monitor farm thing, I'm looking at, I'm going to work with Ben and Richard to try and build in a you know, how we can collect some data, you know, try and understand, you know, maybe earlier is later. Earlier is better than later, that sort of thing. So, if you just click through, I think we just put a couple of things we're going to put on there. Because curb's very good in respect of, you know, there's only limited opportunities you can use it. So, you know, that cost of putting it on now, you've got to foresee over the, you know, your whole crop rotation you're trying to protect sort of thing. So we'll look at building in this curb into the monitor farm thing, which I think will do really well, and we'll do some further observations and that sort of thing, and then we can come back and summarise over the next 3 years sort of thing. So yeah, I'm not sure. Is anyone else?
If you thought about changing the rates, putting half on now when it's a bit dry and half maybe in July, is there something you look in the future? Has anybody done that?
nothing that we've ever done that I can recall. But has anyone thought about that? I've always waited, to be honest. So like in the in the Coxfoot last year, we got to, I think it was the 6th of August, and that was the point. Like we got to, it had to go or it didn't go, and we ended up putting it on, and it still did its job. We were lucky and got rainfall soon after. It was about the only rain, you know, that we got. But Anyone, any comments?
The later you go, though, you get that slack problem with
Yeah.
getting soil coverage.
Yeah.
Well, I read the label again before I covered as opposed to half what you use, but
It's-
they talk more about soil coverage than actually rainfall. And I'm
Yep.
wondering if that goes back to your slide before.
Yep.
And then usually, I'm usually done in May, but it's going to be slow because the plant's small enough and just hope rain's coming.
Yep.
Just using this time of year now, I'd struggle to see enough soil to keep on.
No, even in the, like, I don't know of anyone doing that in clover or any other crops either.
I think where we've got clover and we've got big cover crops, we tend to graze them out first.
Yeah.
So we can get to the ground as part of that process. But going back from my days in the UK when blackgrass and resistant blackgrass was the biggest issue, there's two things that led to poor control of the curve. One of those was deep rooting, so going back to the cultivation thing. Who the director would actually ended up with better grassweed control than conventional
Yeah.
system, and that's part of the depth thing. But the other biggest issue from my memory was if you go later, once that soil temperature warms up again, coming back into the spring, it's the half-life of the curve that gets broken down. Its temperature is, soil temperature is what
Yeah.
microbial activity is what actually breaks the curve.
And the
down. So
plant's growing again, isn't it? So
yeah.
it's also fighting.
But the UK is a bit different. Like cold, wet seasons, you can actually see it in the week following.
What sort of rates are people using on Oilseed rape? Because they'll be higher than the clover rates that we're probably using.
Cheapskates, 1.5 is the minimum for colour, 1.7 if you take here with the bottom, but there's, when you get into tough grass and hot soil, there's no substitute for
Yeah.
plenty. 2.5 for beans, we used to use 3.
Yeah.
So, yeah, definitely buy it, because if you've got issues, you've really got to put it on. And if conditions are against you, you've got to put it on. First rate stuff.
At 3, you could probably cut it in half and do it twice, but at 1.5, probably not.
You're all quite aware it's not cheap.
No. No.
Well, I've quite often taken a different approach where I don't care if it's too dry or not. So that when the temperature is right, it's got to go on.
Yep.
And I've quite often, there's been twice I can recall now, where I've put it on and then it would be anything up to 3 weeks later, you get a rain shower and it'll reactivate. It does sit in the soil and it will reactivate on a rain shower.
Well, that's what, like that picture I showed of the cocksfoot, because that was sitting on the plant. Must have been most of it must have been sitting on the plant to cause the damage, but yet still killed enough of the that was trivialis. Yeah. So like there must be some ability for it to be washed around or something. Yeah.
I'll just make a comment there, Richard. It's not broken down to be a UV. Like obviously it's primary method of degradation is microbial. So if it is sitting on the surface, I mean we do recommend you put it on and wash it afterwards, but it will hang around until there's some rainfall.
Nice.
Are you actually going to do some trials again?
I hadn't considered it, to be honest, until I found some of this data, and then we were thinking, about this presentation, to be honest. We've been doing some in the cocksfoot space, but I hadn't had plans to do any in Oilseed rape. And given what I see there, well, we're probably a year away from, we've sort of missed that early timing now.
It would help if you did it for rape, because FAR gets money out of rape growers.
Definitely. So we'll get whatever we can gather out of the Monitor Farm program, and then that will inform us for next.
Just aside, when we had very mild autumns in the UK, how we used to get around that, we obviously couldn't do it if it was raining, but we'd spike with Frizzlate.
Yep. So
But the old 125 formulation used to be 100, it used to be 300 mils. And
Yep.
what that seemed to do, and we always used to think it was mimic a frost and cold, how it affected the plant, the way it took up stuff. So it could be worth doing. I've actually just done some of that on some carrots to see. That one today actually.
so Fusilade and
Curb, tank mix.
Curb, Fusilade. I'm just trying to think. Fusilade was in that in the data that I presented there. I think there was also what else was in there? There were mixes with with Clethodim. Yep. Yep. So I only pulled out the curb treatments there because nothing actually added to the control over curb on its own. Yep. But if we had the warm weather, maybe they would. Yep. I'm not going to rule out that we aren't going to do any further work because it is a valuable tool, for breaking the hairgrass cycle if we could. We are doing quite a bit of work in hairgrass at the moment, but not necessarily in Oilseed rape.
Good. Was that last one, was it?
Well, we had some stuff to talk about aphids.
Aphids.
Yeah, and then fungal diseases. So, yeah, I'll kick off again and then hand over to Richard. So I've come from a background of biochemistry and plant breeding, so I never thought I'd be an entomologist, but here I am looking at aphids down a microscope every week. But so it's certainly been interesting. So you guys all know why aphids are a problem. They carry viruses. The one that I was talking to a well-known plant pathologist who happens to be my father this weekend. He was talking about a virus, and I can't remember what he called it, but I think it's changed its name, and that's called the turnip mosaic virus these days. I think now it was last year we did we actually had some oilseed rape plants tested from. Did we get them from a variety of different paddocks and stuff? And
3 different. Yeah.
3 different, yeah. So and every plant that we tested showed that there was virus in those plants. Now, those plants were otherwise sort of not too bad health-wise. It's probably most important when they actually were infected with the virus, not whether or not they had it. But it's something that we probably need to do, again, more research on. We don't really know what the yield penalty is for the viruses that they might get. Cereals tend to get a lot of the aphid research. There's much more money coming in from cereals than oilseed rape, but it does cross over, so there's this added benefit. When there's a warm autumn, that is the biggest risk for yield losses due to aphids or to viruses that aphids carry. Because obviously that earlier in the season that they get infected, the more likely they are to actually become stunted and you'll lose your yield. So if it's warm and in the autumn, when the plants are young and there's a lot of aphids around, that's the biggest risk, and that's it's something you need to look out for. We recommend to cereal growers certainly that they monitor their crops to inform spray decisions rather than just go in blindly with an insecticide every time. Partly because of the effect it can have on beneficial insects, which will do the job for you, but also just to save you money as well. So I probably won't talk too much about the beneficial insects today. I think the recommendation from you guys is that an insecticide goes on early in the season as a matter of course. Is that right?
It's probably monitoring to if they are seen, if there's high numbers when we're putting the Clethodim on, we'll put some in.
Yeah, I mean it's cheap enough to throw some in, isn't there, when you're going on with the herbicide anyway. So one thing I can talk about here, if this clicks through. Oh yeah, other thing I wanted to ask. So overseas, there's one of the things that you can do is to plant a variety, but I think in New Zealand we don't really have any particular varieties that would be identified as more resistant to aphids than others, or to the virus than others do. It's just, we don't really have any data on that. Is that right in my thinking there?
No, we haven't tested it for resistant versus non-resistant varieties.
Yeah, that was what I was thinking. Okay. So this is the one I was talking about, the turnip mosaic virus. This is a crop that's been likely infected early and has just become stunted and never really got going as a result. Some more pictures here, which Richard found for me. So this purpling here on the stem, that's a classic symptom of virus infection. Again, purple on the leaf, and some of these stems here almost completely purple, and that's your classic mosaic virus symptoms.
Interestingly, every plant we tested last year actually had virus, even if they were green and showing no symptoms. So like all the commercial plants, and yeah, I don't know how if it's actually going to cause us any issues or whatnot, but something we should just be aware that we've definitely got a lot of virus floating around. Based on the yields we've been achieving, it's not having a massive impact. So how important is it is probably our next real question.
But the other thing it does go to show is that because it's in every plant, that likely also means it's in almost, if not every aphid, because depending on the viruses in the aphids, it's not always the case. You might only get 10% of the aphids that are actually carrying the virus, but if every plant's got it, then maybe every aphid has it too. So has anyone here been to this website, Aphid Chat? It's yeah, one. It's brand new, and we haven't even got a link to it on our website yet, even though it says FAR at the top. If you go to the FAR website, you won't find a link to it, as far as I know. No, it's pretty hidden at the moment, but I have been linking to it from some of our other publications like Crop Action and so on. But this is still in development, although it's pretty close now to what we want it to look like, and it has some good information in it. But this is monitoring data goes here, and that monitoring is all done in cereals. But I think that an aphid is an aphid, and even though they're different species and they carry different viruses, that if there's a lot of aphids in the cereals next door, then there's probably also going to be a lot of aphids in the oilseed rape or in whatever other crop might be around. So if you go there, you can bring up whatever location is nearest to you. I've just chosen Waimate, and it'll tell you how many. I write this little report every week, just saying what I think's going on. It'll tell you how many aphids I found, and if there are any beneficial insects as well, which is probably a less useful number to an oilseed rape grower, because that number is going to vary wildly depending on whether what insecticides have been used, what sort of microclimate is there, what the paddock history is, what other plants, host plants might be nearby, and hedgerows and trees and things like that. So that'll be really different from paddock to paddock, whereas this one will be more consistent across the whole region. And you can see, what a surprise, the aphids have been dropping steadily in the last 2 months. And we're almost at zero now, which would be what would be expected. There's still a few places around South Canterbury that have got a few aphids though, so it's still worth monitoring. It's been warm enough that they haven't completely gone away yet in some places. Pleasant Point was one where I found quite a few today. So this is just some pictures I took from the traps that I put out in. If anyone is interested in what they look like, I've got some traps here that I could pass around, and I had planned to, but we're almost at 4 o'clock already, and I know I've got 2 more speakers, so I'll probably best to just race through. Here's some more pictures of aphids. That doesn't look like flash. This one is actually better than it looks, because almost all of these ones are what we call aphid mummies, which means they are dead husks of aphids which had been parasitized by a wasp. So likely there hadn't been too much insecticide on that one to kill off the wasps. All right, so here's some stuff that Richard, I think, is going to talk about.
I'll just go back one
Sure.
slide, Ben, because what I've found, just observational-wise, looking at crops where I've seen the aphids getting to this point, is when you actually get to the point of looking at them just before harvest, you can tell these aphids that have been affected at the base and in through the stem, and you open them up, and a lot of that seed's actually aborted. So in the past, I think we'd think, oh, okay, it's not got too bad, sort of thing. But you actually look at those pods, and there's barely any seed in it, and the seed's aborted. So I think it's probably something that we need to monitor more closely, and it is knocking a little bit off our yields at the top end, sort of thing. So yeah, I'm not sure. I think would be mostly not too bad in recent years, because the spring has been not too bad. But yeah, in years where we get really dry and they start coming in a lot quicker.
Yeah, I think we are actually losing copying yield with those aphids.
Might also lead in nicely to the next couple of slides, because that feeding damage could actually be a place that Xanthomonas or Pseudomonas could get stuck into the plant as well. And we did the disease survey 2 years ago and found small amounts of bacterial damage to the racemes of oilseed rape. And Mark did make the comment that he wondered if it was a result of something feeding on the plant as opposed to the pathogen itself being the primary source, whether they were secondary infections. So how long do you want us to go, Keith? Like, we've probably already taken up our time.
That slide that shows the percentages of disease.
Yeah, so I just wanted to throw some pictures of Alternaria, because this was the main disease that we found present in the survey. So we did that survey 2 years ago, and this is all the different diseases that Mark could find on the paddocks. So did we end up with 10 or something paddocks? I can't remember. It might have been 10. Oh yeah, 10 sites, 11 different pathogens or present. So the bottom one is the virus that Ben talked about, but in this case, the difference between pathologists, Mark called it beet western yellow virus. Apparently it's the same thing as the turnip mosaic virus. So that's that one. But in the top three here are all Alternaria species, and out of the stems that we had, there's over. Come on. Yeah, come on. Next slide, Ben. Yeah, nearly 70% of the stems that we collected were infected with Alternaria of some description, whether that was on the pods, the racemes, or the main stem itself. So that was the main pathogen that was present, and quite a wee way back for the foma to, or blackleg as it could be at that point where it's on the stem, to actually be the next most prevalent. Let's see, that next one, Ben. So this was the percentage of paddocks that had those diseases present. So 90% of paddocks, there was only one out of the whole lot that didn't have any Alternaria present, and 70%, so really quite low levels of foma, but 70% of paddocks actually showed some sign of it. So what you'd take from that is we're probably doing a reasonable job of controlling foma, which then becomes blackleg. It's just present at a low level in 70% of paddocks. The rest of them are really, they're either there or they're not. So the likes of sclerotinia, it's either in the paddock or it's not, and it's probably more a rotation type thing. Next one, since it doesn't want to change. So I found some trial data again from 2011-12, looking at control of blackleg foma. And I guess this sort of started along with the Alternaria work that we've done to wonder how much infection can these crops actually handle, because there's quite different levels of infection for all of these products in stem canker. So stem canker is where the foma's gone into the stem and caused a lesion that can be the whole thickness of the stem near the base. And it progresses from a leaf infection, which is what we call foma. So this becomes that. But really, at the end of this, they're reasonable yields, and there was no difference between any of those application in terms of seed yield at the end of it. So obviously, the crop can handle a bit of disease. I guess the question is, if you did nothing, how quickly that would build up to become quite a severe infestation over years. So stubble-borne disease will build up over time. So it'd be interesting to repeat that piece of work now that we're 10 years, nearly 12 years further into growing oilseed rape, as to whether the infection levels would be high enough to be more severe. But that's 50% of stems that are black at the bottom if you cut them in half. So there's a pretty severe infection, but there's two untreateds. One of them's almost as good as everything else, or better than anything else in the trial. Go to the next one, Ben. Actually, skip through that one. So this is the black leg as it gets into the stem of the plant, and this is the sort of damage that it can do to the stem once it gets into that point. Go to the next one. And that's the foma on the leaf, and there's another one. Infections. So skip through to the next one. What do we got? And the next one. So this was pictures of Alternaria. So these are the black spots pretty much that appear on the stems and the racemes and the seed pods. So most of the time there's not many other black spots on the stem. So there can be some others on the pods and bits and pieces, but black spots that tend to be on the stem and racemes are Alternarias. Go to the next one, Ben. Whoop. And this is where we get onto the pods. So these black spots, I couldn't actually find a good picture of them. And this one's actually got a wee bit of botrytis on the end of the pod as well. So really, these black spots are what we were looking at in terms of being the most prevalent disease. If you flick to the next one. And on the back of that, we set up this trial actually at Hurst's last year, trying to get some Alternaria under irrigation, thinking that it might have been a dry season. And numerous different products, but again, like this column here, there's not really any difference between any of those fungicide programs, which again raises the question of how much disease can these actually tolerate and still produce quite well. In the literature, Alternaria doesn't tend to take a lot off yield, but we had some pretty severe infestations the year before, where I don't know, Mark was adamant that we should have had yield reductions from having that at the levels of Alternaria that we had. So that's where we went after the disease survey. We didn't get big levels of infestation in here. I think we got through to about 7% pods with the black spots on and the untreated, whereas the year before we were about 50% more than that. So low disease pressure, but where do we go with some of this work other than carry on and see what we can do?
Would it be fair to say there are two different seasons in terms of rainfall?
Very different seasons.
One was a disease builder and one was pretty dry.
One was quite wet versus one that was reasonably dry. Yep, and humidity would have been a lot lower this season. So we tried to pull some products, some timings apart, but it didn't end up going our way last year.
Keith will probably know better than me, but on the firmer thing, I think when we trialled it in 2011 was when we were using things like Astrid, which were relatively good resistance to foam, but the newer varieties, as far as I know, are probably a step change ahead again.
Yep. So we actually have 3 years of FOMA trials, and they all show the same thing. I didn't have time to pull all those together, and that's like, we haven't pulled all that data together, which is a big shame. So it's something that we were talking about last week before last, about part of the program that we'll try and run the next 2 years is to pull all of that data that we've got and put it into a publication. Because there's 3 years of FOMA trials that show no real difference. But it'd be nice to repeat those now and see, because like I said, we're that much further into the pattern of growing oilseed rape that it could be more of an issue. And was it Monday that you actually was brought to our attention that we did a disease survey in 2011 that I didn't even know about. So we've actually got a disease survey from 2011 that we can compare with what we did 2 years ago, which would be a nice comparison to do as well.
Yeah, so we've had I'll just background on the modern farm thing. So we've run that for 3 years. So that's run its course. We're just now setting out with FAR and running it for another 3 years. So as part of that, talking to Richard and the guys at FAR, there's a lot of this information sitting in with FAR that would be good to extract out, and we try and do a crop 101 where we can have all this information sitting there that if there's topics like this come up that we can bring up quickly and have it all in one space sort of thing. So that's going to be one of the goals for the next 3 years. At the end of it, we should have some of this data. Well, probably try and combine all this data that we've got. If there's initially looking at it for the first year, see what holes we've got in it, and then try and fill in some of those spaces. Yeah, because obviously listening to Richard being here, there's a fair bit of information there sitting with VAR that we can extract and get out to the growers.
This is also where I'm pretty sure he had 9 plants tested out of there for virus and they were all positive. But 5 tonne yields out of positive plants raises the question of, while it's positive, is it actually doing us much damage? I guess if they were really severely stunted, then maybe they would be, but obviously, we had big canopies in here, they were quite reasonable plants.
Yeah, my observation is, looking at this sort of data, is there's certain levels of disease in the crop, but different years, something triggers it to express, certain diseases will express themselves more than other years. So sometimes you'll get, like we had a couple of years ago, you'll get a heap of alternaria come through, and then you'll get some years where you won't see much sclerotinia, but the next year all of a sudden there's heaps of sclerotinia in the crops. And we're not really changing the programs up too much, but it may be a timing thing. Or one thing I want to look at this year is late fungicide after flowering. So a November timing sort of thing. And seeing if, obviously alternaria is quite an issue. It's in most of the crops. So seeing if something in a November period will actually clean it up.
I think I only had one more slide of sclerotinia, but we can probably just move on.
Just to see
To
it.
be honest.
Yeah, that's it.
Does anyone want a coffee or a quick drink of water? No? Good. I'll swing into it. Everyone's warm enough? I think they're good. Right. Thank you, guys. So as I briefly explained, the Monterey Farm thing, I think everyone's aware of that, so I won't go into any of those details there. I'll hook into that. So we've been running it for basically 4 seasons now, and this is basically a box and whisker grape, and you'll see our general trend has been upwards over the last 4 years. Previous year was obviously the best year we had, but then we had some of our guys getting quite good yields this previous year. What probably affected it most this past year was the dry, so some crops were able to obviously handle a lot higher and produce a higher yield. Excuse me. We had some of the crops actually run into some moisture deficit, and it did depend on what area you were. So these plots or Monterey Farm paddocks are sort of spread fairly through Timaru, South Timaru, right down to the Waimairi. So we have got a geographical spread in it. So this has been quite interesting, I think. We've been showing this the last couple of years, that we've actually had quite a downward trend in the amount of nitrogen we're using. So in some cases, guys are telling me before they're going in and harvesting it that they're underdoing it, and they're feeling like they haven't got enough, and they're going in harvesting and actually realising that they might not have a big bulky crop, but the crops are actually yielding fairly well. So I think the first year we were averaging about 210 units, I think it was, or 205 units, where last year we were about 175 units. I think the biggest change we have done with the nitrogen is actually changing when we're applying it, so managing it better in terms of looking at those canopies going into the spring and determining whether we need to pump them a bit more or hold back until later to more that green bud stage. So this is a scatter graph of all the nitrogen versus the yields, and as you can see, that's quite a spread. If I actually put a linear line in it, and it actually, as you increase the nitrogen, the yield dropped away. So it goes back to the data that FAR did 2000. They actually did 3 years. This was the third year that they did the nitrogen rate trial, and it's sort of, once you get over that 175, there's no actual yield advantage after that. So this information's been quite interesting to collect over the last 3 years. I think I've been talking a fair bit about it in the last couple of years anyway. And there's a real trend to trying to achieve that green area index going into flowering of 3.5 to 4. So using this model and measuring how much nitrogen we've got in the soil, where the canopies are, we're trying to either push or hold back the canopies to try and hit that target level going into that thing. So going into flowering, sorry. So I think this has been quite successful. I've been just recently reading some information out of Europe. Slightly different system, but it actually is exactly the same, and it lines up with what we're trying to do here. So not sure if anyone had any questions. I think most of you have been reasonably comfortable with what we're trying to target there, sort of thing.
I actually analysed that in a different way to what Keith has done, and come up with a similar sort of break point. So like the previous graph of the nitrogen, we can see yields increasing from a green area index of sort of 1.5 through to about 3.5. It's about 3.5 to 4 where that breaks to be a flat line. So there is something around 3.5 out of there. There's a wee bit of noise about it, but that is sort of the break point of the two lines on a graph, and we could show that at the end if you wanted to.
Yeah. Thanks, Richard. I think what I'd like to do a bit more of is spread this over a wider range. Would that be of interest to growers, if we did it over everyone rather than just the monitor farm stuff, and then you could see where you were compared to other growers? Maybe? Just a thought. Right, soil compaction. So this is another thing I've been looking at. So what I've done here in this graph is we did measurements at different depths, and I've tallied the compaction readings to give us a level of compaction, and then I have put it on a trend against yield, and you can see a general trend. As the compaction levels drop away, you'll get an increase of yields. So this comes back to obviously, rooting depth and how much the plants can access in the soil, sort of thing. So I've got some photos here of just generally what I'm talking about. I think that came up. So as you can see, pretty simple. The deeper the plants can go, the more moisture and nutrients they can access, sort of thing. So I think we've had a general trend of more guys subsoiling going into the crops, and I think it's helping us overall at the other end. So plant numbers. So we do plant number counts every year. Probably what I took out of this information was that there really isn't much difference between 15 and 50 plants per square metre. Oilseed rape, as we probably all know, it compensates fairly well, and the overall, there's not a specific number that we're trying to target, sort of thing. So it doesn't matter if you've got low numbers, high numbers. It always, at the end of it, it seems to compensate pretty well. So here we did some trials with this is actually albina of that heading's wrong. So Albina was a calcium product that they were getting quite good results with in peas, and I thought, well, if they're getting, there could be something in it. We haven't tried the product before, putting it in with, we were putting it in with the fungicides with the flowering. So when it's going through that reproductive period, seeing if a calcium product, which is quite a key nutrient that we don't actually really apply, it generally comes out of the soil. So we tried it over 3 different places, but we didn't end up getting any results. I think a few guys tried it in some other crops. They're definitely getting results in peas, but didn't find anything for this Albina product. So we also tried last year some biostimulants. So I was reading some articles in the UK where they were using amino N going into stem extension, and I thought, well, we'll always have cold soils in that early spring period. We're putting nitrogen on, but we don't know how much is getting in, how long it's getting into the plant. So, a rapidly available nitrogen form, I reckon sounds like a good idea. So I happen to know someone that had a product with that was readily available. So, and she also convinced me that we should be putting on a bioactive, which is a seaweed fertiliser. So we've trialled that as well. So the amino is I'll get Lydia up shortly to explain it better. But it's an organic N, which is readily plant available. So, and then the bioactive, which is a seaweed one. So seaweed's very good on your abiotic stresses. So it's also got quite a few trace elements. So I thought that would be quite good going into the winter to try and keep the plants healthy going through the winter, and then hitting it with this amino, really available nitrogen to kick it into gear into the spring. And this is what we've ended up with in a couple of years of trial. So this bioamino, where we've done bioamino and the bioactive, we've got between, I think it's 250 and 450 kilos a hectare increase, so which is quite significant. So I might get Lydia up now and she can talk a little bit about it. Did anyone have any other questions while she was getting set up?
I think grain sampling through the UTs.
Yeah, just pull that one out. Yeah, well, because I've talked to obviously yourself, Andrew, and then I run into some of the Southland guys that we've got this yen testing done. What I want to do is we've got both plant and seed tests done from a couple of years ago. We did 2 seasons of it. So we've got the information there. I've just got to go in and analyse it really, yeah, and then try and compare that to what we're seeing overseas. Right. So, yeah.
Is there an opportunity to make a consensus? I know we're a bit late for that project we just done. Richard, is there an opportunity to make some grain rapeseeds? Subs and no inputs? You must have to retain samples.
If we've got samples, then there's no reason why we can't.
I know we've been comparing against UK data, but
It's just as interesting, though.
I think it would be.
And I think we've already got 2 years of data.
Yeah.
If not, if not 3, I can't remember back what was happening in the early years here, but there is a couple of years of historical data from those 10 or 12 farms that we could compare with, at least have a little New Zealand database.
I mean, I haven't seen the yen test, but they're just a nutrient breakdown percentage.
The benchmark is interesting, I think, comparing against.
Yes, comparing it against the historical part, it's interesting.
And the removal as well. No, it's actually going to go with grain. The removal.
Have we still got samples from these places? Yeah. Sweet.
And we'll be able to pull up those old tests too.
Yep.
Very good. I'll pass over to you, Lou.
Cool. Thank you for having me here, Keith. It's bring a wife to work day today, so I hope I don't say anything out of line, but I think what I aim to do is just explain some of the muck and mystery around at least bioamino and bioactive, and hopefully at the end of it you've got a clearer idea of why these products got included in the monitor farms, but also why we're seeing the potential results we're seeing. So it's just my interpretation, but Richard and Ben and Keith, you might have some inputs you want to put in along the way as well. So bioamino, what is it and how does it work? So it's 9% organic nitrogen, but 9% at 4 litres a hectare is 0.36 of a unit of nitrogen. So why, as conventional farmers, do we care about that? Well, amino is actually our bioamino is high concentration amino acids. So for the plant to actually be able to metabolize synthetic nitrogen, it takes amino acids to convert that nitrogen and be used within the plant. So what we're trying to do is supply those amino acids, and we're trying to save energy for the plant, and in the process increase nitrogen use efficiency. So it's all around that, getting better results for what we're actually putting on, rather than the 0.36 units of nitrogen. This is an awkward slide, so just bear with me as I'm just using it to set the scene. So why are we applying nitrogen in the first place? We're applying nitrogen to grow the crop, according to Keith. But really, we're trying to build chlorophyll to increase photosynthesis. So we're applying nitrogen to increase those things, but often we put nitrogen on, but we don't see the yield effect that we would expect from the amount of nitrogen we're putting on. That's often a result of many factors. Some of them we can influence, like nutrients, but others abiotic factors, like environmental or water temperature. We can't affect those. So that's where a product like bioamino or biostimulants come into play. So, many amino acids have different roles within the plants, and many amino acid products have different amino acids. So if we just look at our top 3, so this aminogram is an analysis of what's in our bioamino product, the concentration of the different amino acids. So we have glycine, proline, and glutamic acid as our top concentration, I suppose. So if we just keep in mind that we are applying that nitrogen for chlorophyll and photosynthesis and growth, and we have a look over at the functions in the plant of that, is we have the precursor of chlorophyll, a stimulation of photosynthesis and growth stimulation. That's not to mention the chelating, anti-stress, all those other factors that these amino acids are a part of in the plant. Which is probably why in the trial monitor farm trial work last year we didn't see as great a results, because it wasn't a high stress year, whereas this year was a high stress year, which really showed that the difference between the treatments. So this is just a really visual example of how adding an amino acid into the plant might look like. I'm not going to go into that detail. I saw Richard's eyebrows go up there. I'm not going to attempt to even go into this. It's just a visual representation of how nitrogen is metabolized in the plant. I like this diagram because it's got urea getting converted into ammonia, and you can see all the NADPHs and ADPs, the energy that that plant needs to use to then form it into glutamate and those amino acids that we're trying to apply with bioamino. So just a visual of how we're trying to save the plant energy and give it that next step down. So in oilseed rape, when we're wanting early nitrogen to get into the crop, we've got low soil temperatures, low soil mineralization of nitrogen. We're trying to bypass that from a canopy point of view. It's just a soapbox moment that not all amino acids are equal. So On the back of our GLC work we did last year, we had a fish salesperson going around saying that their fish product could be used. It's amino acids. You can use it and expect to get the same sort of results. So as a result, I got our fish boost tested by an aminogram, and that just shows you the difference in the products. Really, fish's fatty acids and proteins, which are much more complex molecules that you're trying to get into the plant. Yeah, and also if you look at it from a nitrogen point of view, if you extrapolate that out to protein, our product is 56% proteins, whereas our fish at 3% is only about 18% protein. So not all amino acids are equal. So our product has seaweed bioactives included in it because Waikaitu makes a seaweed product, but we're also going for the plant stimulation effect of that as well, which leads nicely into the bioactive. So you had bioactive as a singular treatment. We did bioactive and bioactive on one, didn't you? But so seaweed, muck and mystery. So our seaweed is 100% wild harvested fresh brown kelp Undaria, which is a invasive species that smothers the mussel lines in the Marlborough Sounds, so they come along and extract that. So I've just put a couple of graphs there because people often talk about which species are best when it comes to seaweeds or which hemisphere's the better producer of seaweed. This is some independent trial work that we've had done with our product up against other people's products. So we've got Calpac, which has a heap of data behind it out there. I think it's Canadian. And then we've got Africalp, Stimplex, and Puricalp, where in all of them, NZ Bioactive is performing on par, if not slightly better. But the thing for me is that this is a New Zealand produced product, so we don't have to cart it from around the other side of the world. So what do seaweeds do? I think Keith covered that with abiotic stress management. They used to think it was the plant hormones in the seaweed, so the cytokinins and gibberellins and that actually had the effect on the plant, but they've found that if they extract those from the seaweed, it's still having an effect. So they've now come up with a theory that, and proven it, that it's actually through this upregulation of genes leading to a reduction of the abiotic stress symptoms. So what is upregulation? It's just increasing the plant's response to the substance or signal from outside to just boost those cells' specific function. So in other words, it's a signaling compound, and in even other words, it just tells the plant it's not under stress, keep on growing for as long as it can. So you're just extending that growth window out further before you're getting the dry kick in, or that sort of thing. So this is some trial work that we have in cereals, apologies, that just backs up that efficacy in dryland and abiotic stress protection. So we got an extra 1.1 tonne of wheat in dryland, and we got like 0.5% screenings with another wheat variety in dryland also. Keith asked me to talk about quickly the grower leading change work that we were involved with the last 2 seasons. So this is 2 seasons ago's work. It was all based around alternative sources of nitrogen for cereals. We managed to reduce nitrogen by 30% without any significant difference in yield. There was around 0.5 tonne reduction in yield, so for me I still think that's significant. So we're trying to tweak there, but it was quite a obvious trend between all of there's 7 different paddocks, 6 different growers. We had some really high-end users. These are the nitrogen rates down the bottom, the full nitrogen and then the 30%. So we had some guys reducing it by 98 units of nitrogen, and they still got within 30%. One guy had a XP paddock that he put on 400 kilos of urea, and he only got 2 tonne more on compared to his control paddock with no end strip. But yeah, lots of different information there. So this season, the group sort of combined with another group, and then they looked into liquid versus solid nitrogen, and then there was 3 growers that looked at foliar versus solid, and in the foliar one they included 2 litres of amino and 1 litre of bioactive, and they were quite excited with the results, so hopefully next season they're going to look into a few more replicates of that foliar scenario. And then as a result, we took the wheat trial to Calix, got them to replicate it for us, see if we could capture those results in a fully replicated trial. Unfortunately, it was a very late sown wheat paddock, so the yields were down in general, but we were able to maintain with 20% 20% reduction of nitrogen. I was hoping to get a yield improvement, that's why I went with a less reduction this season, but it didn't work. It was such a good growing season that I think if it was a bit more stressed, then obviously it was irrigated. And then the other thing that might be of interest is we've done a kale trial down in Southland this season, where we put our product up against Megafol and Actimax, so an imported biostimulant and a trace element blend, to see if we could compare with that, and looks like we're on par with that as well. So yeah, that's the rest of the range. We won't go there because the bar's waiting.
Very good. Yeah, so from the Manawa Farm things, I'm going to be, encouraging a lot more of this amino N to go on the crops this coming year, because I've seen evidence that it's really kicking into gear sort of thing earlier on, and we'll keep doing some more research and strips and that sort of thing. But I think there's enough there to see, there's some real positive yield gains there, sort of thing. So, did anyone have any quick questions for Lydia on those couple of products, or?
Good staff rates.
Staff rates?
We unfortunately don't sell direct.
Was there any, did you change any of the fungicide, like in that wheat trials? Did you use this fungicide when you were using these products?
No, we haven't tested that one. To be fair, this time round my aim was to put it in with all of the current fungicides to show that there wasn't a disadvantage for including them in their spray programs. That is definitely something I'd like to dig into. FAR did some last season or the season before with a seaweed. Yeah, they did that. Well, I know, I spotted it at their crops day. I'm not sure how that trial ended, but it'd be worth having a look at. I have had growers, my background as a soil consultant, so some of my previous growers had used these products, and we had a bit of a play with fungicide programs where we might have snuck it in instead of when they sort of overlap a little bit. But yeah, no, I don't have any data. I'd like to. Maybe you could include that in your proline trials.
Plenty of research to do. Yeah.
Well, I wanted to, we carry on sort of like the trial work that Keith presented, where we've got some strips and some paddocks, and try and get enough strips and enough paddocks that we get some replication across sites and seasons. It's interesting that we're really looking at it quite early to try and get that early season growth, as opposed to late when you really expect a whole lot of stress. So like applying quite early to try and build that canopy to get it to that green area index that we want, and then hoping that we might get something else out at the end of it. That's for us to try and pull apart as to whether it is actually growing a bigger canopy or whether it's something that's happening later on in the season. It might be that we're seeing something different to what we're actually targeting.
That first year I think you went earlier and later with the bioactive.
Yeah, so you might not have picked up, so when we're putting that amino on is the same time as we're just mixing it with the follicle. So when it's just going into that rapid stem extension period, which is generally sort of in the start of September, so generally soil temperature is still pretty cold.
But generally anything that saves the plant some energy through respiration, like that's why New Zealand's pretty good at growing these crops, is because our our nighttime temperatures are cool. So anything that's going to save us there should allow us to grow more biomass and more dry matter. So if we can save a wee bit of energy, then we should be able to see that in theory come through in terms of yield. Be interesting if we can get multiple years across different soil moisture regimes as well. That would be, we need to talk about soil moisture monitoring in some of these strips, I think.
I've got a South African colleague actually here. I'll throw you under the bus, Voter, but obviously we, in discussions with Voter from where he's from, and sort of western South Africa, you come from quite a dry period place or area, and there was a lot more of these biostimulants and that sort of thing used over there, from what you were telling me, wasn't there?
Yeah, I think we were quite exposed to the elements of irrigation, so quite interesting on the curb topic before as well. Like we use curb completely different to anything that's being done in New Zealand really. But now there's a lot of that kind of stuff going on, just because of the fact that you don't have control over your over the environment and applications. So definitely worth looking into.
Very good, guys. Well, thank you everyone for your attention and coming on. Hopefully there was some information you could pull out of that sort of thing. So, it's been very good. I'd like to thank Lydia and Ben and Richard for coming along, and everyone for coming along, and organised a drink and some food downstairs so we can discuss further if you'd like. Very good, thank you.